This video is learning targets 4 through 7 on the genetics units, unit 7. And for these learning targets, we'll be discussing the idea of protein synthesis. Protein synthesis is the production of proteins in the cell. Proteins are made on the ribosome. Here's a picture of protein synthesis happening. Proteins are made on the ribosome, and ribosomes can be located in two different locations inside the cell. Proteins can be, or ribosomes can be located. Ribosomes can be located in the cytoplasm, and ribosomes can be located on the endoplasmic reticulum. Ribosomes that are located in the cytoplasm, when they make proteins, those proteins are going to stay in the cell. Whereas if the ribosome is made on, or is on the ER when it makes the proteins, those proteins are going to leave the cell. And so some cells are going to make a lot more proteins that stay in, other cells are going to make a lot more proteins that are going to leave, depending on what the purpose of that cell is. Protein synthesis exists in two phases. They are transcription and translation. Transcription is the process of making a messenger RNA from the DNA. And this happens in the nucleus, starting with DNA, going to a messenger RNA. And again, this happens in the nucleus. Think about the word transcription. It comes from the word transcribe. This is taking one form and converting it to another form. So look at this picture. She's listening to music or watching a video and converting it to words, like written words. And so it doesn't change the language, just changes the form of that language. DNA and RNA are the same language. They're both nucleic acids, but it's converting DNA to a messenger RNA. Translation, on the other hand, is taking that messenger RNA and converting it into a protein. This happens, again, on the ribosome. And so this process is taking the language of nucleic acid and converting it into the language of protein, which is basically a bunch of amino acids grouped together. So transcription is where we'll begin. Transcription happens in a number of steps. This is where a complementary strand of RNA is made from the DNA, and this RNA is called a messenger RNA. The messenger RNA serves as a particular code for amino acids, which are in turn used to build proteins. The very first step here is that the enzymes involved in this process have to find a promoter sequence. And this promoter sequence is where the gene begins. This tells the enzymes where to start to the sequence of DNA nucleotides that signal the beginning of the gene. Right? All genes have this promoter sequence. There are a number of enzymes involved in transcription. We're only going to mention a couple. There are different transcription factors involved. One of them is a helicase, and it is going to unzip the DNA, the hydrogen bonds that form between the two strands of DNA. The third step then, most important, is RNA polymerase is going to add RNA nucleotides. So let's think about this for a second. We have a DNA strand, just randomly coming up with these letters, and its complementary strand is T, C, G, A, T, G, G, C, T. Where there's an A, you put T, G, C, C, G, A, T. So one of these strands, they're separating from each other. Now let's say for this RNA, we're going to make a complementary RNA from this bottom strand of DNA. Well, everywhere we see a T, we're still going to use A, C's, G's, G's, C's. But when we see an A, we're no longer going to use T's because there are no T's in RNA. We're going to use U's, uracil. Everything else is going to be the same. We're just not going to use T's anymore because there are no thymines in DNA. And so that complementary strand of RNA is made from the DNA. You can see it on this picture to the right. Complementary RNA is being made from that strand of DNA. And then step 4 then is the process is going to end on a termination sequence. There's a particular sequence of nucleotides that signals the termination of this RNA, and it stops making, and you have this loose RNA molecule. Well, 3 more things need to happen to this RNA molecule before it is final. The first thing is that the introns are removed and the exons are joined together. What are introns and exons? Well, introns are the non-coding portion of the gene, or of the DNA. It doesn't code for anything. So why is it there? Well, the introns can be used, or they're taken out, and then these exons, which are the coding portions or the useful portions of the DNA, can actually be switched around to make alternative proteins. You have like 26,000 genes in your body, but you can make thousands and thousands and thousands more proteins because of the different combinations between the exons. A little bit more than what we need to know in this class. I mainly just want you to understand that the introns are removed, the exons are stitched together, which you can see here in this picture, and again in this picture. The green regions of the exons are left in, the orange regions are taken out. So that was 5, I believe. This is 6. A cap is added to the front end of the RNA molecule, and this cap serves 2 reasons. Just what you would think a hard hat would do. This is a protection, and it also signals the ribosome. Now, we're still in the nucleus. We're not out in the cytoplasm yet, but when this messenger RNA leaves the nucleus, goes to the cytoplasm, this 5' or this cap on the front end is going to signal the ribosome to come. And lastly, well, before it leaves, a poly A tail, poly A as in adenine, is added to the end, and this again serves as a buffer. It serves as a protective mechanism because out in the cytoplasm there are a number of digestive organisms, or not organisms, enzymes that look like this Pac-Man here, and they're desperate to eat these RNAs up. Why? Because your body doesn't want, or your cells don't want lots of RNAs laying around, so the cytoplasm is a way that they are broken down, but you want the important portion of the gene to not be broken down until it's translated, and so this poly A tail is a mechanism against that sort of thing. And then lastly, 8, the messenger RNA leaves the nucleus and goes to the ribosome. So that's it for transcription. Before we talk about translation, we need a couple of terms that are associated with it down. First is, well, what is translation? It's using the code on the messenger RNA to connect the proper sequence of amino acids to make a polypeptide or a protein. 2 interchangeable words there. Here's amino acids. This is the structure of an individual amino acid. Not worried about you reproducing that, but there it is nonetheless. And there are 20 different amino acids found in the human body, all of them differentiated by this R group here. And when those two amino acids, or any two amino acids, are joined together, water is taken out, as you can see here, so it's dehydrated in order that they can be joined together. So this reaction is called a dehydration synthesis. Taking water out to join two things together, dehydration synthesis. And the bond that is made between two amino acids is called a peptide bond, which is why multiple amino acids joined together is called a polypeptide. Another important term that we need to be associated with is codon. A codon is a 3-base or 3-nucleotide unit on the messenger RNA that codes for a particular amino acid. And so there's 3 bases there, GCA, or 3 nucleotides. So 3 nucleotides equals 1 codon, and 1 codon equals 1 amino acids. And so all of your messenger RNAs are in these little groups of 3s called codons. Very important. Well, how do we know what they code for? We use this. Someone figured this out a long time ago, won a Nobel Prize for it. That was good of them, but we don't have to do that. We can use this nice nifty chart. This is a codon chart. We'll take one, AUG, AUG, codes for this particular amino acid. There's another chart that I don't have, and it's a big square chart that we'll learn to use as well, but it's similar. This wheel chart is much easier to use, and so we will use this as well. Let's take another one. UAA. UAA. Stop. What does that mean? Well, there are 3 codons, 1, 2, 3, that are stop codons. These don't code for amino acids. This tells the process of translation to stop. And there is 1 codon that you do need to be familiar with. Here's AUG again. AUG is always the start codon. It doesn't say start on here because AUG does code for a particular amino acid, methionine, and it always codes for that. There's always a methionine at the beginning of every protein. There, you can find methionines in the middle of the protein, but there's always one at the beginning. Another term we need to be familiar with is tRNA, or transfer RNA. Transfer RNA brings the amino acid from the cytoplasm to the ribosome. It also contains an anticodon, which corresponds to a particular codon on the messenger RNA strand. And so each tRNA is only capable of carrying one particular amino acid. So let me explain this. So here's a tRNA. It looks like a T, so that helps. And it connects its amino acid up here at the top. Down at the bottom, you have the anticodon. Means opposite. And so the anticodon is literally opposite of the codon. If the codon is GCC, like you see here, the anticodon is CGG. And it's just part of the RNA. It's not a special structure that it's carrying with it. It's just part of the RNA, but it happens to match up. And no matter what, it's always going to bring this codon, GCC, which is GCC alanine. Every time, it's going to bring alanine with it. Well, here's another example over here. UCG. The anticodon is AGC. UCG is UCG serine. Every time this particular tRNA is going to bring serine. There are 61 different tRNAs out there. 61. Well, there's not 64 because these are 3 are stop codons. But there is a tRNA for every particular kind of amino acid. And so that brings us to translation. We have these nice nifty pictures here, like this one and this one, and they're helpful, but I like to kind of draw my own thing. And so translation, again, what is the process? Well, this is we are making a protein using a messenger RNA, and this takes place on the ribosome. So here's the ribosome. All right. And here's the messenger RNA. So the first step in translation is that the messenger RNA is going to bind to the ribosome. And of course, you'd expect for this first codon to be the start codon, AUG. And then I'm just going to randomly select some letters off the top of my head. There's one set, here's another set, and then here's another set. Okay, the last three weren't random, which we'll see momentarily. All right, so AUG. It has a particular amino acid. That amino acid's methionine. It's going to come down every time. But for the interest of not making this complicated, we're just going to say that this tRNA is bringing these little shapes. Again, these are amino acids, but I'm not going to draw the amino acid structure on there, so I'm just going to draw a blue moon. Now, how does this amino acid know to connect there? Well, it's got this anticodon, UAC. And so UAC is going to come down here. This anticodon is going to come down here and connect. Well, the next step then is another tRNA is going to come, and this one is bringing a, let's see, this one's going to bring a pink heart. And so this pink heart represents an amino acid again, and the anticodon here is going to be GGC because that matches up with the CG here. And so it's going to come and sit here. Now at this point, the ribosome is actually going to shift down. The ribosome does all the moving. The messenger RNA doesn't actually move. The ribosome kind of moves down the messenger RNA. And at this point, the ribosome itself is going to combine these two amino acids. And remember, what is this called? This is called a peptide bond. This is a strong covalent bond. And once that happens, this tRNA is going to go away. And it's going to go back and pick up more blue moons. Well, then another tRNA is going to come down. And you can imagine this whole process happening over and over and over again. But we're going to have a purple horseshoe here. All right, so purple horseshoe, and it's going to be UAA. Where it's anticodon. And the ribosome is going to move down again. This is going to connect that peptide bond once again. And the tRNA is going to go away. And notice our chain's getting longer, and we could keep adding more and more Lucky Charms to this, but that's going to be beside the point because I think you get the idea. The ribosome moves down again, and it just so happens that our last, I think I did that wrong. I did do that wrong. It just so happens that our last codon is a stop codon. Now, again, the stop codon does not code for an amino acid. So when it comes to the stop codon, what happens is this polypeptide will break off and it will fold up. And you can imagine that there's going to be more than 3 amino acids. There's probably going to be, there could be 100, there could be 10,000. And those amino acids are then going to fold up like a protein folds up, and it's going to have a particular job that it does. All right. So if you can imagine this, well, we can just kind of do that here. We can fabricate this and make it really long if we wanted to. You can kind of get the idea. This is going to go on forever and ever and ever until it finally gets to the stop codon and you have this giant protein that has a particular shape and function. And so this is translation. Now lastly, we're going to talk about mutations. Mutations are a change in the DNA, a change in the genetic material. They can be beneficial, they can be harmful, they can even be neutral. That means they don't cause any problem or they don't do anything good. But a mutation is a change in the DNA. Make sure you understand that. A change in the DNA will directly affect a protein, and that protein will affect some sort of body process and usually cause some sort of issue in the body because that protein is no longer acting the way it should, and therefore that process is no longer going to function the way that it should. And so mutations can cause a number of significant problems. All right, so let's look at some of the different kinds of mutations. First, there's a point mutation. A point mutation is a mutation on a single nucleotide. So right here it says CTA, down here it says TTA. That T is a mutation. It should not have been there. It is there now, and so there's going to be a problem. It's going to make a bad protein. What do I mean by that? Oh, here's our friend the codon chart again. And let's just go through some examples here. There are actually three kinds of point mutations. We're going to use this example. Let's say that our DNA strand is A, C, G. Well, that's going to give us an RNA strand of U, G, C, U, G, C. So the amino acid in this case is going to be cysteine, right? So that's if everything's happening as normal. But let's say there's a mutation. And instead of ACG, we have ACA. There's a point mutation. We have a one base that's bad. Well, what's the RNA going to be now? The RNA is going to be UGU. Oh, look, still cysteine. Is this a mutation? Yes, it is a mutation. We have a change in the DNA. Is there a problem as a result of it? No, no change. And so we call this mutation a silent point mutation because it doesn't make a sound. You don't notice it. All right. Well, what if instead of ACG we have ACC? What's going to happen here? UGGs are RNA. UGG gives us UGG. Uh-oh. We have tryptophan. That is not what we intended. You know, if someone tells you the sentence, the dog jumped over the fence, that makes sense. Well, if someone says the dog ham sandwich over the fence, that no longer makes sense because that word has been replaced. And so this, where one amino acid is changed because of a point mutation, we call this a missense mutation. Missense mutation. All right. Last kind of point mutation. Let's say that instead of UCG, we have ACG, we have ACT. That's going to give us a UGA, which is going to in turn give us a stop codon, which is not an amino acid at all. It's an early stop codon, and an early stop codon is like, instead of the dog jumped over the fence, we're going to say the dog, and that's it. We just had an early stop in our sentence. This is a nonsense mutation. Nonsense mutation. Early stop codon caused by a point mutation is called, that's the nonsense mutation. Another type of mutation is called a frame shift mutation. Frame shift mutation results from the insertion or deletion of one or more bases, and this causes our reading frame to shift. Well, what do we mean by reading frame? On the messenger RNA, we read the messenger RNA in particular frames or particular units. These units are codons, groups of 3. We've established that already. Well, if you take one of them out or add one in, then all of the codons are going to shift. So let's look at this picture. Let's look at this middle section, which is the norm. All right, ATG or ATG, CCT, that's the DNA. All right, well, what happens if you insert something after the T. Well, the first couple of codons are okay, but then this second one, which used to be GTT, is now GGT. That second C is, or second T is added here, and notice everything is going to be broken after that. You're going to have a completely different group of amino acids as a result of that. Let's do the opposite. Let's take this A and cut it out. And this is going to move everything this way. And so instead of GGA, now you have GGC, AAT, instead of CAA, AAT, ACT, instead of TAC. And so all of these amino acids that as a result are going to be completely busted because of that shift in the reading frame. So that can be a particularly devastating type of mutation. There are two chromosomal mutations I want to mention, inversion and translocation. Inversion is when portion of the, or multiple portions of the chromosome are inverted or backwards. You can kind of see here in this one, there was a section of the chromosome that was ABC and DEF. Well, if you'll notice, this section here has become inverted. And so now BC is backwards on this side, and now DE is backwards on this side, and the whole thing is inverted, and the whole thing is broke. Nothing's going to work the way that it should, and so this would be a massive mutation on that whole chromosome. Same thing with the translocation. We have portions of the chromosome that relocate to another portion or swap, and so ABCDEF becomes ABCJKL GHIDEF. So two chromosomes are now completely ruined because of this translocation mutation. Again, these are chromosomal mutations because they take place in the whole chromosome. And so the question is, are mutations harmful or beneficial? Well, yes, it can be both. All traits are, if you trace any trait, it goes back to a mutation because all traits are derived from an adaptation which has evolved over time, which was originally some sort of mutation in a species. So in one sense, those mutations were good. Well, we have mutations today that are bad. Mutations cause cancer. Mutations cause diseases like Tay-Sachs and cystic fibrosis. Very bad things. But there are beneficial examples of mutations as well. Sickle cell anemia is a good example of this. Sickle cell anemia is not beneficial if you're from this country, so it's a bad mutation because in this country, if we get a disease like malaria, for instance, we can just go to the hospital and get fixed. But if you live in Africa or if you live in Southeast Asia where malaria is particularly devastating, if you have sickle cell anemia, it can actually be beneficial because individuals that have sickle cell anemia are immune to malaria. So that's a good thing. Also, polyploidy in plants. Plants have more than one chromosome in a pair. That can be a very beneficial thing for plants as they're more hardy and able to withstand more.